Welcome to XenoTech's webinar, Suicide by Binding, Putting Time-Dependent Inhibition of CYP Enzymes into Perspective. I am pleased to introduce today's presenter, Dr. Brian Ogilvie. Brian received his Ph.D. in toxicology from the University of Kansas Medical Center. He has been with XenoTech since 1997 and currently serves as a Vice President of Consulting. Brian is an author or co-author on over 50 scientific posters, peer-reviewed publications, or book chapters on the topics of drug metabolism, transport, and drug-drug interactions, and has represented the company as an invited speaker at various drug metabolism conferences. As Vice President of Scientific Consulting, Dr. Ogilvie is additionally involved in quality management and strategic planning for the company in scientifically relevant areas. It is my pleasure to present to you Dr. Brian Ogilvie.
Thanks, Michael, for the introduction. So I wanted to give you a little bit of background about the title. I was having some conversations over the last few years with clients or other people who were saying, you know, it looks like we have a time-dependent inhibitor, maybe even a mechanism-based inhibitor, which is sometimes referred to as a suicide substrate. And, you know, looking further, it turned out that in some of these cases, the potency of the inactivation really did not rise to a level where there would need to even be a drug-drug interaction study performed. And so I sort of came up with this as a, not really a cautionary tale, because certainly irreversible inhibition can kill a drug, especially if you have other backup drugs that have similar properties. But in some cases, it doesn't really mean the death knell for that compound. So I'm going to go through briefly just to define the types of time-dependent inhibition and their importance, the in vitro approaches to evaluate the various types of time-dependent inhibition. So that includes screening versus our definitive studies, and then approaches to predicting clinically relevant DDIs from time-dependent inhibition. For case examples, I have a couple of interesting ones. Drugs that only cause time-dependent inhibition in vitro, or what we would refer to as a system-dependent effect. And then some that do cause clinically relevant time-dependent inhibition. Most of these are older drugs, but I have one I can mention that is recently approved in 2018. So first of all, time-dependent inhibition of CYP enzymes, what it is not. TDI of CYPs is not simple direct or reversible inhibition, but there can certainly be both. Reversible inhibition is, of course, most often competitive and can cause clinically significant pharmacokinetic drug interactions. I'm sure you're aware. And this is a case, just simply put, where the inhibitor and substrate both bind to the same side of the enzyme. The inhibitor in this case is often a substrate that simply has higher affinity. And so the effect is reversible and decreases as the inhibitor drug is cleared. And if you're looking at it in vitro, competitive inhibition can be overcome by increasing the substrate concentration, sort of the hallmark of competitive inhibition. There are other types of direct inhibition. I'm not going to go into those today. So more definitions here. Time-dependent inhibition of CYP enzymes, of course, can cause clinically significant pharmacokinetic drug-drug interactions that are often of greater clinical concern than the direct inhibition because of the potentially long-lasting effects. So even after the drug that causes time-dependent inhibition is withdrawn, that effect can persist for quite some time. And if a xenobiotic inhibits P450s in a time-dependent manner, it's typically metabolized. In other words, it usually is also a metabolism-dependent inhibitor too. And in fact, we frequently use that term. However, I've noticed that over many years, most regulatory agencies just simply default to time-dependent inhibition without regard to whether or not there's metabolism involved. So different species can form. If it's a more potent direct inhibitor than the parent, you have what we term a reversible time-dependent inhibitor. So many examples of this, I highlighted are fluoxetine, mainly because S-fluoxetine is a different case, as you'll see in a minute. Amiodarone has a number of metabolites, and it can get quite complex with that. We'll go into that today either. Additional definitions. So 2, you have a compound or a species that does not leave the active site and binds irreversibly at or near the active site. This is often referred to as irreversible time-dependent inhibition or mechanism-based inhibition. That particular phrase I don't think a lot of people like, because I think most most types of inhibition would have a mechanism or a suicide substrate, and you'll see this often in biochemistry text and papers. Examples here include mebefridil, which I'll talk a little bit about, mifepristone, and I'll talk about gemfibrozil glucuronide as well. There's various subtypes of this. So you can have heme destruction, apoprotein adduction, and other types that I won't go into great detail today. Most of these you'll see are subtypes, sub-conversations where I could have another hour worth of conversation on. So my apologies if I'm sort of glossing over some of these. Third type, and I'll cover this again to some extent later on, is when you have a species that coordinates very tightly to the heme iron of the cytochrome P450. So this is actually termed quasi-irreversible inhibition, and it's due to the formation of a metabolite inhibitory complex. So examples here include some macrolides, such as erythromycin and clarithromycin, as I mentioned earlier, the S enantiomer of fluoxetine. And this is essentially irreversible over the time frame that an enzyme exists. So in other words, irreversible inhibition persists until new enzyme is synthesized regardless of the mechanism. But again, irreversible time-dependent inhibition or metabolism-dependent inhibition may not be a showstopper for a lead candidate, especially if you don't have a good backup, especially if it happens to be one that cures an acute cancer or something like that. On the surface, if you're looking at the clinical data, it can actually be difficult to see the difference between time-dependent and direct inhibition. So here's ketoconazole, a very potent direct inhibitor. In fact, it actually binds very tightly to the active site, but can be reversed. And it increases midazolam AUC by about 15-fold. This is one of the seminal papers from 1999 showing this. It was, of course, known beforehand. But around the same time, Jana Beckman's group published this in 1999 and showed that mebefridil, which is now known to be an irreversible time-dependent inhibitor, increases triazolam AUC by about 9-fold. Within 10 months of mebefridil being approved, it was actually withdrawn from the market because of this potent inactivation of CYP3A4. There are many factors that played into this. Part of it is that it was a calcium channel blocker. There were other calcium channel blockers, and so physicians would often switch patients from one calcium channel blocker to another to find out which one would work best for that particular patient. And a lot of calcium channel blockers like nifedipine and others happen to be CYP3A4 substrates. So even when you withdraw mebefridil, it has a very long half-life and it has a very long duration of inactivation of CYP3A4. And you put that patient on nifedipine, they would then have a very high level of nifedipine or other CYP3A4 substrates. Now I'm going to move on to the in vitro approaches. And this will get a little bit tedious, but I'll try to go through it pretty quickly, defining the screening versus definitive studies. The high throughput screens typically focus only on direct inhibition of P450s. And if you look at this paper published in 2009, it was Nature Biotech paper. This looked at 17,000 compounds. Each one of these hexagons represents similar compounds. And it's a heat map of inhibition toward each of these enzymes. But within this paper, it quotes, as no pre-incubation was included, this database will miss mechanism-based inhibitors. And that's out of 17,000 compounds. So Alfred Zimmerlin's group out of Novartis published in 2011 and noted that this error was widely recognized in the mid-2000s, saying that although reversible inhibition testing is well established for predicting drug-drug interaction potential of clinical candidates, the time-dependent inhibition has become the focus of drug designers only recently. So again, the mid-2000s when people really started to pay attention to this in screening. Most contemporary CYP inhibition screens actually do incorporate evaluation of time-dependent inhibition. And if you consider the IC50 shift assay is a very popular example in high throughput screening, it's simply an extension of that. It just adds NADPH during a pre-incubation. And you don't have to have the full 7 concentrations. You can do this at maybe 3 or 4 concentrations. I've seen multiple setups for those high throughput screening assays. But if you look at the definitive studies that we do typically, so we'll have, in this example, human liver microsomes with ketoconazole. We have a 0 and a 30-minute pre-incubation with an NADPH. And we'll see that the IC50 actually increases slightly after that 30-minute pre-incubation. Therefore, we can conclude that that's direct inhibition. I mentioned mebeferdil before. In this sort of screen, we would see IC50 shift of, approaching 90-fold. And clearly, there's a time-dependent inhibitor there with mebeferdil in the human liver microsomes. So single concentration high throughput screens have also been developed to rank order time-dependent inhibition. And again, going to Alfred Zimmerland's group at Novartis, they used 10 micromolar concentrations for greater than 4,000 drugs and some new chemical entities with 0 and 30-minute pre-incubation. This would approximate a rate of inactivation, what's called the K observed, in units of reciprocal minutes. And the positive negative cutoff value is set at a K observed of 0.02 minutes, reciprocal minutes. So if you look at this graph, this is the cutoff value. These are the 4,000 compounds, whether they're drugs or NCEs. The drugs are represented by triangles. And you'll note that once you get over this cutoff, you'll see drugs like omeprazole, ethinylestradiol, sorafenib, and on up to really high levels of inactivation rates. So you get up to imprinavir, trilliandamycin, raloxifene. They have quite high rates of inactivation. Interestingly, from the study, 71% of registered drugs that were flagged as time-dependent inhibitors in this assay were not flagged at all in the direct inhibition screen. So again, pointing out the fact that you could have a clean bill of health, so to speak, in your screen, and then miss completely the time-dependent inhibition. So once people started realizing that, then the focus was on, okay, let's throw out anything that shows a time-dependent inhibition signal. So now I'm going to move on to definitive direct and time-dependent IC50 shift assays. So our definitive measures within one assay, direct, in other words, a 0-minute pre-incubation, time-dependent inhibition control. So this is a 30-minute pre-incubation without NADPH, just to make sure there's nothing that happens during that 30-minute pre-incubation without NADPH. There could be another enzyme involved that actually activates the enzyme that doesn't require NADPH. There could be some slow binding inhibition. There could be simply, glutathione addicts that are formed, et cetera. And then the time-dependent inhibition, 30 minutes with NADPH. And so you'll see an example here, published in 2011, where I used omeprazole. And with the 0-minute pre-incubation, I have an IC50 value of around 7. It actually increases slightly without NADPH and drops to about 2 micromolar, or approximately a 4-fold shift to the left, showing that we do have time-dependent inhibition of CYP2C19 with omeprazole. So controls also include, obviously, the solvent control, positive control inhibitors for both direct and time-dependent inhibition. And the marker substrate is set at Km, or the S50 for the reaction. And what this allows you to do then is take that IC50 and very easily extrapolate with the Chung-Prustoff equation to the Ki value. And in fact, we published on this in 2015. Lois Haupt is first author on that paper. Human liver microsome concentrations are set at less than 0.1 milligram per milliliter to minimize partitioning into those membranes and protein. And then the substrate incubation should be short, 5-minute, relative to the pre-incubation of 30 minutes. So even though we have this result with this 4-fold shift, we still don't know if this is irreversible time-dependent inhibition. So the question becomes, where do you go next? So what we typically do now is, and have done this for at least 12 years or so, is look at time-dependent inhibition reversibility by ultracentrifugation. So this provides qualitative answers to provide better decision-making. So one concentration of drug is chosen from the IC50 shift assay, and it's pre-incubated with NADPH. Substrate here is 10 km to minimize any direct inhibition, and in the human liver microsomes at a low concentration, and incubate for 30 to 120 minutes, depending on the shift that we observed. After that pre-incubation, the samples are then ultracentrifuged to remove the unbound drug and any metabolites that may have formed. And we typically have 4 groups of samples, solvent control, and then a group where the compound is pre-incubated only. So this replicates what we saw in the IC50 shift. And then pre-incubated with microsomes that are reisolated. And I'll show you an example of these data here in a minute. And then finally, samples that are pre-incubated, the microsomes are treated with a very high concentration of potassium ferrocyanide, which is known to dissociate the quasi-irreversible metabolite inhibitory complex prior to centrifugation. So when we see ultracentrifugation does not reverse time-dependent inhibition, we can conclude we have irreversible time-dependent inhibition of CYP3A4. So in this case, I'm going to show you mebifidil at 2 micromolar. Again, there's the IC50 shift, and we picked around 2 micromolar, where we have a nice strong effect after 30 minutes. And in this case, what we're seeing, solvent control has essentially no effect after 30 minutes. These are rates of testosterone 6-beta hydroxylation, a marker of CYP3A4. We have mebifidil before centrifugation. We see that strong time-dependent inhibition. This is mebifidil after centrifugation, and you can see the 30-minute pre-incubation, there's not a reversal. And even in the presence of potassium ferrocyanide, there's still not really any significant reversal. So therefore, we can conclude that this is, in fact, an irreversible time-dependent or metabolism-dependent inhibitor of CYP3A4. So when we have ultracentrifugation that does not reverse time-dependent inhibition except in the presence of potassium ferrocyanide, we would have quasi-irreversible time-dependent inhibition. And I'll show you the example of troliandomycin at 25 micromolar. This is an interesting case with MICs because you can actually visualize these on a UV vis spec. And over time, you'll see this peak forming at 455 nanometers, whereas P450 obviously is at 450 nanometers. That increases over time. You can actually measure the rate of inactivation by how quickly this peak appears. But the easier way to look at this in terms of irreversibility is, again, this ultracentrifugation method. And we see, again, solvent does not much. TAO before centrifugation mimics the IC50 shift. After centrifugation is not reversed. And if we stop there, we might conclude that this is simply an irreversible inhibitor. But with potassium ferrocyanide, you see that it is reversed, and now we know we have a quasi-irreversible inhibitor. And if we were to go back after the fact, if this were a test article, we might be able to see this UV vis spec with a peak at 455 nanometers. So another interesting example is when you have opposite effects on CYP2C19 time-dependent inhibition by omeprazole enantiomers. So this is work I performed around 2015, and showing this data a little bit differently. So I've got pre-spin, post-spin, and then post-spin plus potassium ferrocyanide. But again, you can see the solvent control is essentially doing nothing. R-omeprazole, we see some direct inhibition. After the centrifugation, both are reversed. Omeprazole, we see less reversal after spin, whether we have ferrocyanide or not. But then you see even less reversal with esomeprazole. So what this tells us is that esomeprazole is an irreversible time-dependent inhibitor of CYP2C19. R-omeprazole, however, is a reversible time-dependent inhibitor. So if these were new drug candidates and the R enantiomer had activity, it may allow for an internal decision to prioritize it over the other enantiomer. So for definitive K-enact and KI determinations for irreversible time-dependent inhibition, these provide quantitative answers to put the irreversible time-dependent inhibition into a clinical context. And typically we have 5 inhibitor concentrations plus a solvent control. You can use 4 to 5 per incubation time points. And these should be performed at saturating marker substrate concentrations, so 10 times the km if solubility permits, and this minimizes any residual direct competitive inhibition. It also usually incorporates a 10 to 30-fold dilution factor to decrease the remaining test drug and metabolite concentrations so that all you're seeing is irreversible inhibition. And these values, the K-enact and the KI values, can be used in the basic mechanistic static or mechanistic dynamic models, in other words, PBPK, to predict the clinical impact on a co-administered drug. So one example of this, again, going back to omeprazole, what I did here is I used concentrations ranging from 5 micromolar up to 60 micromolar, pre-incubation time points up to 10 minutes, because I knew from preliminary experiments this happened relatively rapidly. And what you'll see are these slopes of these lines actually represent a rate of inactivation. And you plot those rates on a direct plot, analogous to a Km and Vmax plot. And what you get is a Ki value, which is analogous to a Km. In this case, it's the concentration that represents half of the maximal rate of inactivation, which is the Kinact, which shows us that at 0.046 per minute, about 4.6% of CYP2C19 would be inactivated per minute at saturated concentrations of the inhibitor. And so now I'll go on to how to predict clinically relevant DDIs from time-dependent inhibition. I won't focus in great detail on this. This is the basic interpretation of the final FDA guidance released earlier this year, where you have your K observed and you put in your K inact and your Ki value unbound, so it's corrected for the binding. You have this 50-fold safety factor built into it. You take your unbound Cmax and your unbound Ki value in vitro, and the cutoff value then is greater than 1.25. If it's less than that, then you can conclude that you don't have enough inactivation to warrant a clinical drug-drug interaction. It's often overpredicted, and so you may have to go to a mechanistic static model, which the FDA also has within their guidance. I won't cover that now. But I will cover an example of a mechanistic dynamic or PBPK modeling. So this is a simulation of time-dependent changes in the active CYP2C19 level after 14 days of omeprazole being dosed at 40 milligrams twice a day. So it's a rather high dose, but not something that's, out of bounds of clinical use. SimSimp simulator was used. This was version 10.2 because it was back in 2011 to assess time-varying effect of repeated omeprazole administration on active CYP2C19 over two weeks. And what we see then is this over 2 week period, you can see the active CYP2C19 fluctuates as you go over time. Now, if we change that to a lower Ki value or a higher Kinact value, which I did obtain under different conditions, you would see more of an effect. But I felt like this one really represented closer to the truth, because when you look at the simulated increase in S-methanotonin ADC of 1.5-fold increase, it was similar to the 2.2-fold increase observed in the 2C19 substrate moclobamide upon omeprazole co-administration. So now I'm going to go on to case examples. So either drugs that only cause time-dependent in vitro, could be an artifact of the in vitro system, or it could simply be a system-dependent effect. In other words, you need to choose the right test system to get the right answer. So first, I'll focus on inhibition of CYP3A4 by ezetimibe in human liver microsomes. And when you look at this by itself, you see this 100-fold shift in IC50 for a 30-minute pre-incubation with an ADPH. So it looks like a pretty potent time-dependent inhibitor. And so it is an effective time-dependent inhibitor, but it does not cause clinically significant inhibition of CYP3A4. So why is that? So looking a little bit further, we used the ultracentrifugation method, and we found that ezetimibe minus an ADPH, of course, does nothing. With an ADPH and after ultracentrifugation, we don't see reversal. And with terracyanide, we don't see reversal either. We know we don't have a quasi-irreversible inhibitor. This is a true irreversible inhibitor of CYP3A4. So we know that, and it still does not cause clinically significant inhibition. So why is that? So we changed systems. We went to human hepatocytes, and in this case, we actually see activation of CYP3A4 with midazolam, which is not uncommon. You'll see that with midazolam in particular in my experience. But the point is, there's no inhibition in hepatocytes. So that fits with the lack of clinical CYP3A4 inhibition. even though it's a potent metabolism-dependent inhibitor of 3 or 4 in human liver microsomes. But in hepatocytes, the hypothesis is that glucuronidation protects CYP3A4, consistent with what happens clinically, where the glucuronide is actually the major metabolite. In fact, you see relatively little circulating ezetimibe, and I'll summarize this at the end with one other example that includes structures. So we'll come back to that at the very end. So another case where you have drugs that only cause time-dependent inhibition in vitro. This is either system-dependent effect or simply an artifact, and we actually looked into this in quite a number of ways and published this just a couple years ago in Drug Metabolism Disposition. But what we found, and I'm just showing one example of a phosphorothioate oligonucleotide, we see inhibition of CYP1A2 and actually a little bit of time-dependent inhibition of CYP2C8. So we looked at this compared with hepatocytes. So just like ezetimibe, we changed systems, and what we see there is that there are essentially no inhibition in hepatocytes with CYP1A2, whereas we do see it in human liver microsomes, nice IC50 value. With 2C8, we see some inhibition in hepatocytes, IC50 about 70 micromolar, human liver microsomes only about 12 micromolar. So definitely a difference between systems here. And similar results were actually obtained recently with the investigational oligonucleotide, which is called Imeltastat. Faraz Kazmi and his colleagues at Janssen published this just last year. And they used only cryopreserved hepatocytes and saw very little inhibition in cryopreserved hepatocytes. And that is also a phosphorothioate backbone oligonucleotide. Finally, drugs that can cause clinically relevant time-dependent inhibition. So these are approved marketed drugs, and gemfibrozil, I published this in 2006. This is a case where gemfibrozil itself inhibits CYP2C8 with relatively high IC50 values, 120, actually going up to 150 micromolar after 30 minutes of pre-incubation. But if you take the glucuronide, now you see an IC50 towards CYP2C8 of 24 micromolar, drops to 1.8 micromolar after 30 minutes of pre-incubation. And the reason we started looking at this was because there were about 100 patients that were killed by the interaction with cerivastatin, which is a CYP2C8 substrate. And cerivastatin, not gemfibrozil, was withdrawn in 2001. And looking a little bit further, what we found, the acylglucuronide actually inactivates CYP2C8 relatively efficiently. I ran this over a range of concentrations you see here, and at 2 different conditions, so either a low protein or a high protein. It did change the Ki value, but the Kinact value was essentially the same. And gemfibrozil is still marketed. Now, the labeling states that it's a strong CYP2C8 inhibitor, and dosing reductions of CYP2C8 substrates may be required. So this is an example where you can simply manage this through labeling. Another approved marketed drug that causes clinically relevant time-dependent inhibition is esomeprazole. And actually, its major metabolites also irreversibly inhibit CYP2C19. I'm not going to show you all the data that I used to prove that it is irreversible. But if you look at the Ki and Kinact value, and if you take Kinact, divide it by the Ki, and this is in units of reciprocal minutes per millimolar, reciprocal millimolar, so you have to multiply these by 1,000. What you'll see is that esomeprazole itself has a value about 15. So that's a pretty decent efficiency of inactivation of CYP2C19. And when you couple that with its 2 metabolites, you now have a case where esomeprazole in particular can inactivate CYP2C19 relatively efficiently. And yet it's still marketed, in fact, over the counter. The prescription strengths do have labeling stating that Clopidogrel is metabolized to its active metabolite in part by CYP2C19 and avoid concomitant administration. This is actually an interesting example because Clopidogrel is a prodrug and actually has to be activated, as it says, by CYP2C19. So this is a case where irreversible inhibition actually leads to less efficacy of the victim drug whereas normally you would see an increased concentration of the parent and therefore more toxicity or adverse events. In Clopidogrel, I mentioned that previously is also an unusual case. Like gemfibrozil, it's actually converted to an acylglucuronide that causes potent, irreversible, time-dependent CYP2C8 also. So this was reported by Jana Backman's group in 2014. And again, they have these nice K-enact plots, get a K-enact of 0.047 per minute and a Ki of 9.9. So this is a pretty efficient inactivator of CYP2C8. So Clopidogrel is, of course, still a marketed, widely used drug. And 2019 labeling actually notes that the acyl beta-glucuronide is a metabolite of Clopidogrel is a strong inhibitor of CYP2C8. And that Clopidogrel can actually increase systemic exposure to drugs that are primarily cleared by CYP2C8, thereby needing dosing adjustment and appropriate monitoring. Now, sorry, let me back up a step. So these are all old drugs, right? So it may not have been picked up at the time they're going through development. There's at least one example I know of that's been approved in the last couple of years. This is Duvelisib, and it's indicated for multiple types of leukemia and lymphoma. And if you look at the approval package, it's interesting because the Ki and K-enact for CYP3A4, they don't define the conditions, but it has a Ki of about 3.8 micromolar and a K-enact of about 0.15 per minute, which is pretty high. And if you look at that K-enact over Ki value, that comes out to about 39, which is a pretty efficient inactivator of CYP3A4, and yet it was approved. So in other words, contemporarily, you can put a drug through an NDA, and especially if it's indicated for something like cancer or you have unmet medical need, then it's not going to be a showstopper. So going back to gemfibrozil and ezetimibe, I just want to summarize this quickly with the structure. Considering phase 2 metabolism in some cases and opposite effects of glucuronidation. So I mentioned gemfibrozil and Clopidogrel as well. Both have these acyl groups where you get this acyl glucuronide forming, and gemfibrozil causes significant time-dependent inhibition of CYP2C8, but not in human liver microsomes by itself. So that's a false negative. Then we look at ezetimibe. Anyone who's used to looking at structures will notice that phenolic group. It's going to be a prime site for phenolic glucuronidation, and it happens very rapidly in this case. So although it's a good time-dependent inhibitor of CYP3A4 in human liver microsomes, that 100-fold shift in IC50 does not cause clinically significant inhibition of CYP3A4. So this is an example of an in vitro false positive in human liver microsomes. So in the case of gemfibrozil, the activation to the glucuronide actually causes irreversible time-dependent inhibition of CYP2C8, and the opposite with CYP3A4. So one thing that you could do here, if you have a case like this, is either add UDPGA to the human liver microsomes or run something like ezetimibe in hepatocytes, or even gemfibrozil. I actually did this, didn't show the data. If you take gemfibrozil, put it into hepatocytes, you will see the inactivation of 2C8 because you give time for the glucuronide to form. So finally, this is an interesting case. Irreversible inhibition can actually be a benefit. So Kobusastat, which is an approved drug with no pharmacological action, and it's marketed as a single formulation as Tybos, approved in 2014. This is not an anti-HIV drug. It's used only to boost the exposure of other anti-HIV drugs listed here. And within the label, it says the mechanism of action is a mechanism-based inhibitor of cytochrome 3A4. And the indication also is a CYP3A4 inhibitor indicated to increase systemic exposure of atazanavir or darunavir in once-daily dosing regimen in combination with other antiretroviral agents in treatment of HIV-1 infection in adults. So just to show you how potent this is of an activator, it increases midazolam AUC by 1,800% after about 2 weeks of dosing. And so this was a strategy that was used in the past. So if you take ritonavir, for example, one of the early anti-HIV drugs, it does have activity as an antiretroviral, but it also inactivates CYP3A4, and that was used to boost the pharmacokinetics of these other antivirals. So in conclusion, both CYP inhibition screening and definitive studies for candidate drugs have become sophisticated in their ability to detect time-dependent inhibition over the years. And then I showed you methods for discerning reversible from irreversible time-dependent inhibition, as well as some basic approaches to put the in vitro results into context. And basically, the take-home message is simply because the signal for time-dependent inhibition is found, even if it's later shown to be irreversible, does not necessarily mean it's the end of the road for these suicide substrates. Modeling may provide evidence that such inhibition is unlikely to be clinically relevant, or the data could be an in vitro artifact upon further investigation. I provided examples of approved marketed drugs that cause irreversible inhibition, and this inhibition can be managed through labeling changes and is not typically a contemporary reason for withdrawal from the market. Now, some of the questions that I've had in the past, of course, there are examples of cases where you have covalent binding to the active site of the drug. So like quinolinic acid is often brought up. It's an inhibitor of CYP2C9. And in that case, you can actually have an immune response. So there can be, in some cases, a correlation with toxicity. But in my experience, that's relatively rare, and it depends on the exact mechanism of the irreversible inhibition. So anyway, with that final comment, I'll take questions, and I appreciate your attention.
Thank you, Brian. While we wait for questions, I wanted to mention the information about our services and test systems, published papers, posters, other webinars, and more are available on our website. If you would like to be notified of future webinars, you can submit a webinar topic request form or register for our newsletter. I should note that this link will be changing in the next few weeks as we launch our new website, but this form should forward to the new form. If you have any further questions regarding Brian's presentation or any of Sekisui XenoTech's contract research services and products, please feel free to ask them in the post-webinar survey or contact us through our website. For today's Q&A, Brian will be joined by Lois Haupt, a principal scientist in our program oversight department with 20 years of experience at Sekisui XenoTech. Brian and Lois, feel free to unmute and start answering questions whenever you're ready.
Okay, thanks, Michael.
Thank you, Michael.
So one question I have here is, how do we control for microsomal viability in a 120-minute incubation? I can say that we typically will go out even to 4 or 6 hours with microsomes on occasion. Of course, you do, in fact, lose some activity. There's a natural rate of decline that seems to be specific per enzyme. And so you can account for that by taking that slope, if you will, for the solvent control. We typically don't do that because in our experience, it's minimal and it just adds a lot more to the experiment. But if you actually do want to, we certainly can. And you get that rate and you simply add that into the rate of inactivation overall for the enzyme.
Okay, the next question is, can you modify the method for irreversible TDI determination for compounds that are autoactivator at low concentrations and inhibitor at higher concentrations? So when we're designing the follow-up TDI experiments, we will focus generally on the part of the IC50 curve, the IC50 shift, where we actually see that time-dependent inhibition, because so we want to focus in on that area to optimize the measurement of inactivation. So we typically won't focus so much on the activating any concentrations that are causing activation. We'll focus on the part that's actually demonstrating the time-dependent inhibition.
Okay, and then there's another question here about for time-dependent reversibility, about ultracentrifugation. What if the compound is tightly bound with the enzyme, like what we saw for plasma binding fraction unbound measurements? So that's a good point, and you may not entirely be able to account for that, especially with drugs like clotrimazole, possibly even itraconazole or ketoconazole to some extent. However, we've shown that ketoconazole, we can reverse it after ultracentrifugation, and we will actually rinse the pellet a number of times before spinning it back down and measuring residual activity. So ideally it works in, I would say, 90+% of the cases. There may be a case or two where we have an artifact, but hopefully that would bear out when you go to do the Ki-K-enact. Afterwards, you would see very non-Michaelis-Menten type kinetics in that case.
Okay, so the next question is, in cases where a glucuronide conjugate is a TDI of CYP, can the microsome assay be run with UDPGA and alomethicin? And yeah, that is something that can be done, and I believe Brian has published that method in his gemfibrozil publication.
Thanks. There's another question here about, for hepatocyte inhibition, was binding corrected, and do you know intercellular concentration? So that is always a real sticky wicket. There are several methods to determine the intracellular concentration in hepatocytes. I'm not particularly a big fan of any one of them over another. I think they all have limitations. Of course, something might be better than nothing, it could be argued. One thing that was done in the gemfibrozil paper is I had the in vivo data from rats that was provided by a Japanese author whose name escapes me, and I apologize for that. So when you have data like that, you can use that to extrapolate, but I agree it's not perfect if you don't account for intercellular concentration. But that, again, is one of those more difficult things to approach. Sorry, there's another question here on the oligo number 3. What's the explanation for potent time-dependent inhibition in human liver microsomes, but no inactivation in human hepatocytes that have good passive permeability? So we did quite a bit of work, and if you look at the paper, you'll see more detail. We even went so far as to try to filter out the oligo prep. We thought maybe there was some residual compounds that were inhibitory that just simply weren't getting into hepatocytes. We even looked at perforating the hepatocyte membrane with meninsin, nigiricin, and adding back in an NADPH. And as I recall, we could then recapitulate that inhibition. So in other words, the oligo was being kept out of the hepatocytes. And unless you have an antisense oligo that is targeted for a hepatic indication or target, that generally would be the case with a phosphorothioate, not necessarily with other modalities. But again, that's something that I don't remember all the details, but if you look at the paper, especially the last one with the meltastat, it does go into more detail on that. Okay, there's another question here on, for early discovery screening, would you recommend screening with HLM plus UDPGA to avoid false positives? I would probably say for early screening, if you can incorporate that into your program and you have a lot of compounds within a series that would be glucuronidated, that could be useful. But then again, we've only seen a handful of examples, and it does seem to be towards CYP2C8, where a glucuronide, especially an acylglucuronide, will inactivate. So that, I think, is very programmed to specific type of decision.
Okay, and then there's a question on, please comment on the pros and cons of supplementing in vitro TDI assays with superoxide dismutase or catalase. That is something that we have discussed in the past when we've had particular compounds that we thought it might be beneficial. But every time we kind of discussed it, we actually just came back and ran the assay in hepatocytes, because the hepatocytes, the question becomes, what concentrations do you supplement into the microsomal test system? And if you incubate with the hepatocytes, it has everything already there.
Okay, and there's a question here about, why do we think trilliandomycin did not fully inactivate CYP3A4? It seemed to only reach about 80% inhibition. In that case, we actually specifically picked a concentration that wasn't near complete inhibition. We've had problems in the past where if we actually choose a high concentration that we know will completely inactivate, the question becomes now, so did we just simply not see any activity because of an analytical issue? It's preferable to at least have some activity present, and then you can see the effect of reversal when you put in the potassium ferrocyanide in that case. Potentially we could go to 90–95% inhibition. It's just a compromise in that case.
And so then we have a question: at what stage of drug discovery would you investigate mechanism of TDI using the ultracentrifugation method? That's ultimately a call for your company to make. We have a lot of clients that wait until the definitive assays, until we're providing, we're conducting studies that are meant for regulatory submission. But if you feel that it's important enough to do early on, that might be something you need to consider as well.
Is the only reason for significant IC50 differences between the 30 minutes without ADPH and 30 minutes with an ADPH due to non-CYP metabolism? I think that may be the main reason, but then in other cases, and I wish I could remember this paper, I just, I've seen it recently and haven't had a chance to read it completely, but the title certainly caught my eye about atypical time-dependent inhibition that occurred in the absence of an ADPH. I don't know what the mechanism was in that case. I believe that it was mentioned something about glutathione adduct being formed, and it could be that it's a molecular acceptor and binds to part of the enzyme. There could be other reasons. You simply have a slow binding increase in inhibition. And then I've also seen cases with even omeprazole to some extent, or other sulfur bearing compounds, that there will be a slight difference between the 0 and 30 minute where the IC50 value either goes up or down slightly. And it's just another control that we include. I mean, ideally what you would do is you would do like the ultracentrifugation method for everything, right? And not worry about this minus NADPH. You would just get rid of the compound completely so you know that it's irreversible. But for the IC50 shift experiment, we're not using a dilution. That is basically just a compromise to look for the non-NADPH dependent effect that might occur on rare occasions. And then what's the relationship of time-dependent inhibition with metabolism and chirality? Can we say that time-dependent inhibition will not occur if there's no significant metabolites or chiral moiety formed greater than 10%? I think it depends on the potency of the inactivation. So for instance, if you have one enantiomer that's only present at 10% of the other one, but it simply has a higher potency and efficacy in terms of inactivating the enzyme, then that could be more clinically relevant than the other enantiomer. So one of those, it depends questions.
So one of the questions is, do you typically measure fraction unbound in your DDI, IC50 shift, or KI, K-inact assays? We don't typically measure that. We do try to keep the microsomal protein concentration as low as possible, around 0.1 mg per mil, to correct or to try to minimize any non-specific binding to the microsomal protein. However, the FDA does acknowledge in the most recent guidance that you can use calculations to adjust. You can calculate the theoretical fraction unbound using calculations that have been published. So we certainly can do that as well.
Thanks, Lois. I have another question here about, is there a correlation of time-dependent inhibition and hepatotoxicity? So this is one thing I stayed away from, because there certainly are examples where when you have true covalent binding, you can actually have antigen formation. So tenelic acid is one example that's been widely used. There are certainly others where you do get antibodies that form to particular P450s and other liver proteins, but that doesn't always occur. And so I don't know necessarily what the correlation of it is. It can be a signal to look for something else. And so I think if you have taken together some signals in the clinical study that there may be some, say, increase in liver enzymes, et cetera, that you need to look at that more closely. And in part 2, does the rate of enzyme synthesis increase to compensate for time-dependent inhibition? I'm not aware of that. I think it can occur in certain cases if it's prolonged. But if you look at the modeling, you simply have the rate of enzyme synthesis that has also degradation that has been determined through a variety of approaches. And that's about the best we can do under most cases for the model. The modeling, when it comes to PBPK, of course, has to be validated and recover what would be a clinically relevant effect. So you can't simply go in there with an assumption and then come out with a model and say it's good. You have to do some validation of that.
So I guess the next question was, in the examples you have picked for approved drugs that show TDI, is it correct that the inhibition was detected after market release? Do you believe that it would be possible today to get a drug approved with TDI? And I believe Brian pretty much covered that in the talk, that, yeah, TDI is not necessarily a roadblock to getting a drug approved. There have been drugs that have been approved despite having caused time-dependent inhibition. And as far as gemfibrozil, and that was something that was noted after the drug had been approved.
Yeah, thanks, Lois. And then another question, technical, for KI, KNACT, is it the larger number that indicates greater risk of clinically relevant DDIs? So this is another one of those it depends answers, but generally speaking, I think that if you have a much higher KNACT, in other words, a much more rapid rate of inactivation. So once you get up to, values that are like 0.7 per minute, in theory, what that means is at saturating concentrations, you're inactivating 70% of the enzyme per minute. But then it also comes into the Ki value comes into play, which is analogous in this case to a Km. So if looking at the Ki and KNACT, you need to look at both of those components, and that's the efficiency of inactivation. There are ways to rank order those with other compounds where those have been described. Think about something like mebafidil probably at the top, other compounds, and then see where your compound fits in terms of its Ki over KNACT value. And that would be one way to address that issue.
So the next question is, how best to evaluate a drug with very low via CYP clearance in vitro for TDI, extend the pre-incubation time? So the thing about CYP inhibition, the CYP inhibition assays, is that you don't have to have a lot of metabolism to see an impact, or a lot of metabolism of the drug to see an inhibitory effect. If you think about it, we're using very low protein concentrations. We're using a 30-minute pre-incubation time. Most of the drugs that we're testing have much longer half-lives and take a lot more to metabolize them. So one of the things that we do, if we see an IC50 shift and it's not a very significant shift, we can extend the pre-incubation time when it comes to designing follow-up experiments, and that usually is helpful. We typically try to avoid increasing pre-incubation protein concentrations just because that can defeat the purpose due to additional microsomal protein binding.
A question now is time-dependent inhibition of drugs. So if autoinhibition can have an impact on exposure following multiple dose. Can I elaborate on this based on experience with the case study, if I recall? So certainly autoinhibition has an impact on the exposure following multiple dose. So if you look at drugs like mebafidil, and it's largely stated that mebafidil was withdrawn because it was just another calcium channel blocker, but it happened to cause this prolonged inactivation of CYP3A4. It actually has to do with a long half-life of the drug itself, and the fact that people were often being switched from one calcium channel blocker to another, which most of them happen to be CYP3A4 substrates. So then you go in with nifedipine or another drug, a day or two later, and you still have CYP3A4 knocked out. So when you have autoinhibition, you see that signal, and you certainly would have for something like Reversible, and you have accumulation, then that's a signal for time-dependent inhibition. And you can certainly have impact over multiple doses. And in fact, it will take, in certain cases of victim drugs, depending on its half-life, many half-lives to reach a new steady state, at which point you may be into the toxic range.
Okay, so for mechanistic TDI studies, KNI or KNAC, do you apply statistical analysis when comparing K-observed values for the low efficiency time-dependent inhibitors? So we do review the data, ensure that everything is, the inactivation is following a first order kinetics. So we do review that. We also look at the standard errors to make sure that everything makes sense with that. We don't calculate a p-value or anything that kind of statistical analysis.
Okay, and then a few other questions here. Have we had any experience differentiating between CYP3A4 and 2J2 time-dependent inhibition given the overlapping substrate specificities? We did actually develop a CYP2J2 assay several years ago and looked at differences there with a more selective substrate. And I know that there certainly were reports. We actually tried to obtain samples of some of these mechanism-based inhibitors of CYP2J2, but in typical practice, most clients do not want to look at CYP2J2. And I think that's largely because it's simply not in the guidance as being something that you have to do and maybe a nice to have in certain circumstances. There would certainly be cases where, if I had a compound that looked like, I don't know, terfenadone analog, where I would suspect that 2J2, CYP3A4 overlap, I might recommend it, but those are few and far between. And then another question is, for compounds where glucuronidation is protecting against time-dependent inhibition in hepatocytes versus human liver microsomes, could you simply add EDPGA to the HLM incubation? And the answer is yes. In fact, I did that with gemfibrozil glucuronide, and I believe there's another example that has been, we actually did that internally with ezetimibe. I didn't show those results here, but I believe we may have put that in a poster at one point or another.
Is it mandated? So the next question, or one of the next questions is it mandatory to discern reversible from irreversible time-dependent inhibition prior to determining KI and KNAC? I wouldn't say that it's mandatory, but it is scientifically advised. We do want to make sure that any inhibition that we're seeing is not reversible, because with the design of the KNAC and KI experiments, we're using a really high substrate concentration, and that's going to counteract any reversible inhibition. So if we don't establish irreversibility prior to running the KNAC and KI experiment, we won't know that if we don't see enough inhibition in the KNAC KI experiment, we don't know if we need to optimize those assay conditions, or if it's because the inhibition that we were seeing was not truly inactivating the enzyme.
This is a question on, I understand glucuronidation can make different or opposite impacts on time-dependent inhibition. So if TDI is exclusively on CYP3A4 without any other effects, is TDI potency the same between human liver microsomes and human hepatocytes? Some papers indicate the difference between human liver microsomes and hepatocytes. Why is that? Any insights will be very helpful. So I think that depends. I would look at the structure first and anything else that I might know about the compound. So if I look at the structure and it's pretty clear that you've got something like a phenolic group that's going to be rapidly glucuronidated, that tends to be a giveaway. Acyl groups that can be form an acyl glucuronide. But if you have in vitro metabolism data, or you have data showing what the major metabolites are, that can certainly be a big clue that would inform that decision and why there would be differences between hepatocytes and human liver microsomes. Another case would be the oligonucleotides that I mentioned, where I didn't show this, but in hepatocytes with imeltastat, that was the publication from Faraz Kazmi out of Janssen last year. They used only hepatocytes and see very little inhibition. And so that's a case where it's essentially an in vitro artifact looking in microsomes versus hepatocytes, where these particular oligonucleotides don't end up in the hepatocyte to any great extent.
Okay, so I think I can take the next couple questions. Going back to calculating the question fraction unbound in incubations, doesn't the recent guidance require unbound Ki? So what do you use for fraction unbound? And then how do we calculate Ki with IC50 value? So if you're running your IC50 experiments at the Km for the marker substrate, you can use an equation called the Chang-Prusov equation. And if you make the assumption that there's competitive inhibition, the IC50 value, or the Ki value, is the IC50 value divided by 2. Now, so the recent guidance does acknowledge that this is acceptable way to kind of calculate that Ki value if you from the IC50 value. As far as using the unbound Ki value, yes, the guidance does mention using the unbound Ki value. However, it does acknowledge that you can calculate the fraction unbound using equations, using certain equations, and so you can adjust the Ki value that's been experimentally determined or calculated from the IC50 value using that theoretical protein, the theoretical fraction unbound.
And I think we're coming up on the hour, so I know we didn't answer all the questions, and we'll try to get back to individuals after the fact for ones that we did not answer, and our apologies for not getting to those.
Yep, this wraps up today's session. Please stick around for the post-webinar survey, and thank you for taking time out of your day to join us. Have a great rest of your day.